So the outline of the presentation is here. I'm going to talk about immunotherapy for the treatment of cancer, and really some of the clinicians are calling it a game changer, and I want you to make your own judgment there. I'm going to talk about immunogenic cell death, which is a way of killing cancer cells that can engage the immune system. I think it's a special way of killing cancer cells. We'll talk about copper and copper complexes and the fact that I think they can define a new class of agents for treating cancer. And that's linked to some of the more interesting new data coming from other groups talking about Cuproptosis, which is another way of killing cancer cells. I'm going to talk about a formulation that's being developed that is one that we believe is going to achieve tumor vascular normalization, and then ask the question about whether this is actually going to be beneficial in the context of immunotherapy. And then, as Gilbert just mentioned, most of the last 15 years, I've been focusing on proactively developing combinations. And it is really based off of this idea that cancers at this time are always treated with multiple therapeutic agents. And I don't understand why, in the context of a drug development program, you're developing drugs as single agents. There are some reasons for that, but I think it's a mistake, and I think it needs to change, and I'll talk a little bit about that as well. So I think people are, those that are in the oncology area will be aware of the hallmarks of cancer. This was Hanahan and Weinberg that developed this. It started off, I think, with 8 hallmarks, and then it expanded to 10, and then this individual, Sylvia, expanded it further. But it really talks about some of the primary targets that people look at when they're trying to treat cancer. And the ones that people are familiar with are in this red circle. Hopefully people can see my arrow move. So the top red circle, sustaining proliferative signals. Those are your standard anti-cancer drugs that hit rapidly dividing cells. We always are worried about the protective mechanisms that cells develop that cause them to resist death. And can you overcome that resistance mechanism? And then a newer one, I'm going to say new in the last 10 years, is the idea that cancers have developed a way of avoiding immune destruction. And if you can overcome those ways of avoiding immune destruction, then you can allow the immune cell itself to recognize the tumor cell and achieve a therapeutic response. And that really is the area that we're going to be talking about today. So when one thinks of immunotherapy, there's a whole bunch of activities going on. I've circled some of the activities that I hope people are aware of. So in the bottom part, this cancer cell killing really is the checkpoint inhibitors, where you have antibodies that are targeting PD-L1 proteins to achieve improved therapeutic responses. There are dendritic cells. This is the one that's labeled up above too. Dendritic cells that present antigens to T cells, and that can actually have a therapeutic impact. There are other antibodies like the anti-CTLA4 antibody, or cytokines like IL-2 or IL-12 that can promote T cell priming and activation and immune recognition. And there's other ones that are looking at things like CAR T cells, cells that are isolated from patients, that are modified and then re-injected back into the patients for therapeutic response. All these strategies have been turned into therapeutics that are making a difference for people with cancer today. So we talk about CAR T cells, there's Chimera, and that's targeting CD19 cells in leukemia and lymphoma. The cytokines, the IL-12 story is an old story, but it's an interesting one. I always encourage people to look at that one. Checkpoint inhibitors, there's a number of them available. They're active against the multiple cancer types. And again, I'm going to talk a little bit more about the checkpoint inhibitors. And then this DC-based vaccine for the treatment of metastatic prostate cancer. So the idea that immune evasion is a critical feature in oncology is being addressed today by approved drugs that are overcoming that immune evasion strategy. And our question is really whether we can do something that can actually improve the activity of some of these immunotherapeutics. So in that context, the areas that we're looking at, primarily 2 areas. One is this ICD thing called immunogenic cell death, and I'll go into a little bit more detail about immunogenic cell death. And the other one is vascular normalization. And I'm going to talk a little bit about vascular normalization. And then I get really curious, and this really has to do with one of the graduate students working in my group, which is asking the question about what if you use something that could achieve vascular normalization at the same time as achieving immunogenic cell death? Could that be the trick to augmenting immune responses to existing immunotherapies? So is it a game changer? I think it is. And I stole this particular figure from the literature, from the internet. And it really is trying to highlight what immunotherapy is doing to oncology, regardless of the indication. So you'll think of immunotherapeutics being used in melanoma or in lung cancer, but it's being used in lots of different therapeutic indications. And what one notices, if you start looking at all the different indications, that almost invariably, when you use immunotherapy, you see this plateau effect, this area where you now have long-term overall survival. And that was different than what people saw before when they used conventional therapies, which increased lifespan, but eventually the patients would succumb to the disease. So this idea that you're creating overall improvements in survival is surprising, game-changing, and important. And when I think of overall survival, this curve is going out to 3 years. I think the oncologist will always say it's if you have patients surviving for 5 years or longer, that's a pretty big achievement. Giving you a real life example, and I'm going to spend some time on this particular story. I didn't start my stopwatch, so somebody has to tell me when I talk too much. I'm going to give you a story about metastatic bladder cancer, and the story goes in the context of I wanted to get funding from the Canadian Institutes of Health Research for support of this program. And first time I submitted it, there was concerns about the fact that I was sort of generic in terms of the cancer. They really wanted me to specify an indication. And at that stage, I started looking at indications. Our group was particularly interested in colorectal cancer, but there were other groups that I was working with that were interested in ovarian cancer or lung cancer. And that was, again, a perfect opportunity for me to say, let's see if we talk to clinicians, what would they say? And I talked to a clinician at BC Cancer, and one of the indications that he brought up, this is Bernie Eigel at BC Cancer, and he brought up bladder cancer, and I really never gave any consideration to bladder cancer. That conversation started 9 months ago, and I got really excited because it was pretty simple idea. Bladder cancer patients that are metastatic are invariably treated with cisplatin. When they're given the cisplatin, if they don't respond or if they progress, then they're given a checkpoint inhibitor, period. That's what was done. So I was writing a grant and doing that thing, and then personal things got in my way, and I didn't submit the grant. I resubmitted it just recently for the March deadline. Prior to that, I reached out to Bernie and said, are you willing to help on this grant? Would you be willing to participate on this grant? And he goes, we need to talk. And I go, uh-oh, something's happened. And I did talk to him, and he brought up this study. And he said, the standard that I was talking about 9 months ago is changing, and that there's going to be a new standard probably 6 months to a year from now. So when you develop a research program, you need to think about what the standard of care is and how you might impact that standard of care. So I think it's an important message that people always, if they're looking at development of a therapeutic, that they are talking to clinicians about what they need. I'll always encourage that. He referred to this study, and I forgot to put the date on this study. This was published in JCO in 2023. It just came out. And it talks about an antibody drug conjugate. The antibody targets nectin on the surface of cells, and the toxin is a zinc alkaloid toxin, which is very, very potent. And it's combined with Keytruda, okay, I can't say the word, this checkpoint inhibitor, and it's being used for a bladder cancer, advanced metastatic bladder cancer. And the outcomes were being measured in terms of duration of response, which is A, progression-free survival, which was B, and overall survival, which was C. And you can see that the results are actually pretty spectacular if you compare it to just cisplatin. I don't show that data on here, but compared to just cisplatin combinations, this outcome was quite remarkable. And this was being done in the context of patients that had never seen cisplatin. So all the toxicities associated with this nasty cytotoxic drug were being avoided, but you're getting improvements in therapeutic response. This is now in a phase 3 clinical study. It's ongoing. It is going to define the new standard of care, and that's what Bernie brought up to my attention. What was really interesting about this particular study is that the strategy was to combine an antibody drug conjugate that induces immunogenic cell death, which is exactly what we were talking about, with a checkpoint inhibitor. And what we're sort of asking is, can we define a best-in-class ICD inducer, or can we define novel ICD inducers, which would be maybe better in terms of overcoming any resistance to prior treatments? We're not the only people doing this. There's a whole bunch of clinical trials that are ongoing. This is just looking at lung cancer, but if you look at all other indications, I think last time I counted it, there was actually more than 1,000 clinical trials looking at strategies to improve checkpoint inhibitors when used in combination with various agents. So it's a hugely competitive, exciting area that is having a huge impact on treatment of patients with cancer. And I've been asking this simple question: if we're getting a 20% or a 30% improvement in overall survival, can we double that? Can we get now 40 or 60% improvements in overall survival? And just as a reminder to the audience, I do submit grants, and sometimes those grants are reviewed. And one of the comments I got on one of the submissions was, oh, that's not really significant. And I would argue that till I'm blue in the face, that if we doubled the overall survival in the context of bladder cancer, that would be a huge benefit to that patient population. So I think it's a lofty goal, and it's one that we really should be pursuing. So trying to improve overall survival, it's a complicated area. It involves a number of different attributes of the cancer. I think it's been summarized most simply as this idea of a cold tumor, which is immunosuppressive, versus a hot tumor, which is immune supportive. And when you get a therapeutic response, it is because you're dealing with a hot tumor, one that is immunosupportive. When you're not getting a therapeutic effect, it's because you're dealing with a cold tumor, which is immunosuppressive. And the question is, can we convert a cold tumor into a hot tumor? And this is a general question being asked by many, many different investigators around the world. Our interest, as I just mentioned, was in this context of the induction of immunogenic cell death, and also tumor vascular normalization, and perhaps the combination of the two. So I don't have a good transition here. What does copper do, and how does it fit in this whole story? Well, copper is an interesting therapeutic strategy, I guess, in general. So people have been talking about copper chelation for therapeutic benefit. People have been talking about copper supplementation for therapeutic benefit. And then, of course, if you're looking online, if you do Facebook, you'll often see advertisements for these lovely copper bands. These copper bands, I hope everybody in the audience is wearing them, because it is supposed to energize you and make you more excited and increase all sorts of health benefits. So I don't have any interest in copper bands. You could buy them from any number of different sources, but highly recommended. So how did we get into copper? It's an old story, and I'm going to talk about this old formulation that we developed starting at BC Cancer, and it really talked about the first generation drug combination product for the treatment of cancer. Now that story started off with the idea of just taking liposome composition 1, mixing it with a liposome composition 2, and putting it in a single vial and treating patients with that single vial. The other concept was this complex concept where we co-formulated the two drugs in the same liposome. This B strategy here, co-formulation, was the strategy that was pursued in the context of the company we started called Cellator. It was deemed to be regulatory easier than the other strategy. So this is the one we ended up pursuing. All along the line, and one of the other talks I give is really this talk that I've learned through my whole life is I'm constantly criticized, I'm constantly told you can't do this. And one of the things when we formed Celator was, you'll never be able to do this. You just have to develop drugs one at a time. That's just the way it is. Don't bother even trying. But Celator did this, and I'm going to talk a little bit about one of their stories here. And it is based off of this ratiometric dosing. And the product that has received approval, this is an old slide because it talks about a phase 3 clinical trial, but it is now approved for use as CPX351 or Vyxeos. And it's a combination of cytarabine and daunorubicin, co-formulated in a liposome and used for the treatment of AML. If you're going to develop something and you want to see it having some value in a cancer patient, you need to be able to protect the technology. It's an awkward conversation because we should be publishing our data. It's a scientific question, but frankly, our science is being geared towards development of improved treatments. If we can't protect those treatments along its development pathway, then it's not going to be developed. When Celator started, we needed to develop an IP strategy, and it came out of the assays that we were doing. And the assay that we were doing was this assay of drug-drug synergy. And it was a simple assay. You looked at the single agent activity, I'll call it single agent A, and you looked at single agent B, and then you looked at the different combination of A and B at different ratios. And that was guided by people that developed this median effect principle, Chow Talali. And you can look at any number of ratios. We looked at 3. And when you did a cytotoxicity assay, what you discovered is that some ratios, the 2 drugs were very antagonistic. They didn't work well together. At other ratios, the same 2 drugs were highly synergistic. And when you look at a cytotoxicity curve like this, you can't make any heads or tails of it because it shows log concentration. But when you look at combinations, you don't know which drug you're looking at. So there's a software that's available, and it's online. I think it's called CompuSyn, but it used to be called CalcuSyn. And it's a analysis software that you can plug in your data, and it can analyze it by producing something called a combination index, the CI value, and it defines a number. That CI value could be 1. It says the 2 drugs are working well together. It's additive. That's a good thing. But those 2 drugs, if they're not working well together, could be antagonistic. And that's a bad thing. You know, you're not taking advantage of the drugs. Surprisingly, 2 drugs at a different ratio would be synergistic. And the argument, this is not using any drug carriers, it was just free drugs. But the argument was, if these 2 drugs are needing to be synergistic at a defined ratio, then after administration, you need to be able to be sure that that drug-drug ratio is being maintained over time. That was the basis of the intellectual property that was developed. And I think drug carriers are the only way that one can actually look at maintaining drug-drug ratio over time. I won't go through the in vitro data for CPX351, but the optimal ratio that was defined across a number of different cell lines was this 5-to-1 molar ratio of cytarabine and daunorubicin. The formulation, I'm not describing this, but cytarabine was passively trapped in a liposome. So as you're making the liposome, cytarabine was trapped inside. Daunorubicin was encapsulated through a remote loading process, which involved copper complexation. So daunorubicin is a drug that binds copper, and you could add daunorubicin to the outside of these copper-containing liposomes, and it would be moved from the outside of the liposome and get inside as it formed a complex with copper. By changing the lipid compositions, you could eventually define a formulation that following administration could maintain this 5-to-1 molar ratio over time in vivo. And that had never been done before. And we did this for a number of different drug combinations, but this is the cytarabine daunorubicin story. So you look at the elimination as a function of time in animals. So this is mice after IV injection. And you can see in this inset curve here that the 5-to-1 drug-to-drug ratio was being maintained for at least 24 hours. The lower figure is preclinical evidence that this was therapeutically beneficial. So these diamonds that are filled is the combination of the 2 drugs in 1 liposome, the co-formulated product, where you're seeing 90% overall survival. If you looked at the optimal combination of the free drugs, you got about, I don't know, 23% survival. You looked at the individual liposomal formulations, you saw therapeutic benefits over the control, these circles, but clearly the best therapeutic result was achieved with the co-formulated product. What you need to recognize about this co-formulated product is that the dose that was being used of the drugs was far less than the dose that was being used when you combine the free drug. So there was a 50-fold higher dose of cytarabine when using the free drug, and there was a 2-fold higher dose of daunorubicin when using the free drug. This is a classic example of synergy. Less drug, improved therapeutic activity. And that was pretty exciting. And that led to a clinical study which showed data like this. There's been other clinical studies showing this treatment where you compare the same 2 drugs, and this is cytarabine and daunorubicin given the 7+3 standard of care treatment in comparison to the CPX 351 formulation. And you can see in the red line that you're seeing that vast improvement in therapeutic activity. This moved on to phase 3 clinical studies and eventually led to approval by the FDA of the product Vyxeos, which is CPX 351. And it was a copper-based formulation. At that stage, copper was being used to help formulate the drug. We never gave any consideration to the fact that copper might be therapeutically important. I do put the dates on this slide just so people get a sense of how long this takes. And this was a very fast process. This is one where the first preclinical studies were being contemplated in 2003. And then the actual approval happened in 2017. So it's about, depending on how you count it, 10 to 13 years to get approval. So as I was mentioning, that formulation, the Vyxeos formulation, contains copper. I don't think anybody really gave much consideration to copper, but copper is becoming a more interesting agent these days. And it really is around this discovery, I think it was made a couple of years ago, of Cuproptosis, which is a copper-dependent cell death. And it is based off of, if you get a lot of copper inside your cell, it can actually cause proteins to aggregate, and this creates a proteotoxic environment and leads to cell death. We never really gave that much thought, but as you'll see in a few moments, we really have been talking now about the potential that copper may be an important therapeutic activity, a therapeutic agent. On its own, that's good. And I'm going to do this transition to disulfiram plus copper. It's a link to activities that we've been doing in our lab for some length of time, but this is a recent publication that came out in 2022, which looked at disulfiram in the presence of copper in the context of this thing called immunogenic cell death. And immunogenic cell death is measured in vitro by certain parameters, these DAMP markers. And those markers include release of HMGB1, expression and mobilization of calreticulin onto the surface of the cell, which is due to endoplasmic reticulum stress, and release of ATP from the cells under situations where the cells are still alive and viable. And this changes the environment within the tumor such that the tumor now is more easily recognized by immune cells within that tumor. And that can actually enhance cancer cell death mediated by those immune cells. This is really the concept of immunogenic cell death. I don't want to develop a product that is solely focused on immunogenic cell death, but focusing on a compound that is inducing immunogenic cell death to augment the activity of an existing immunotherapy is really quite interesting. So let's talk a little bit about the disulfiram story, because for me, this is a recent publication, but it's an old story. And I hope all these slides work because they're fully animated by one of my lovely graduate students, Devin, and I try to unanimate them, but let's see how this goes. The story around disulfiram is a bit of a long one. It goes back to the 1900s, and it goes back there in many different ways. It was used as a vulcanizing agent in the creation of tires. But one of the things that they noticed for people in those factories was that they developed alcohol sensitivity. So if they drank, they became very sick while working in this environment. And it was because of this idea that disulfiram inhibits aldehyde dehydrogenase 2, which is important for the metabolism of ethanol. So if you can't metabolize ethanol, you get buildup of certain byproducts, which lead over to this hangover effect, makes you very sick. That information was used to create a drug for alcohol abuse. It's called Antabuse. It's Disulfiram pills, 500 milligrams. And if you take these pills and you have a drinking issue, you will get very, very sick. So that was the basis of this. And this was approved in 1951. Since that time, it's been used for a whole bunch of different things to treat bowel cancer in mice. There was a case report in human studies where you saw regression of established breast cancer. There was a phase 2 study run in Europe in breast cancer. Whoops, that's my animation thing kicking in. There was a phase 2 study with HIV. I became interested in sort of like 2018, 2019, because that was a time when lots of people were screening for activity of drugs against cancer stem cells. And one of the hits that multiple groups identified was disulfiram. So we became very interested in disulfiram. I got a graduate student, and that graduate student was assigned the task of developing disulfiram for treatment of cancer. He, a knowledgeable individual, Moebi, told me I was full of something. He basically said disulfiram is an agent which is not stable. It's rapidly metabolized to this compound called dithiocarbamate, DDC. That's further metabolized to other compounds. And that DDC, it's a known agent that binds copper. It was used in the early 1900s to actually measure copper levels in water by just adding DDC to the water. And you can now measure the formation of this copper complex, this copper DDC complex. That copper DDC complex is absolutely water insoluble. But what was clear from the literature, it was this compound, this copper DDC, which was a therapeutically active, interesting agent. Because of that, there was a whole bunch of people that started working with disulfiram in mice cells, liposomes, or PLGA nanocapsules. And then our group was trying to develop what we thought was the active agent, which was copper DDC in liposome. And whoa, this was going out of control, sorry. And there was another group that was developing it in the context of BSA. So the strategy that we started off with was based off of what we described with Vyxeos. If you put copper inside a liposome and you add a copper complexing agent to the outside of the liposome, the agent will go through the membrane, bind copper, and become trapped inside the liposome. And that's what Mo did. Again, we published this some time ago. And it was a very simple reaction. It was quite efficient. We could measure the amount and the efficiency of loading based off of this relationship of one copper binding two DDC molecules. When we did our therapeutic activity, we saw therapeutic effects. So there was a delay in tumor growth relative to our controls, and that's shown here. And we have stars showing that this is statistically important. But it was progressive disease. And normally when I look at things that are progressive disease, it's not that exciting. So we sort of took a breath and stepped away from it. 1 year later comes out this Nature paper. So annoying. And they talked about copper DDC using copper chloride, not copper sulfate, to form the complex in the presence of BSA. They published this in Nature. They did see therapeutic activity. They didn't call it limited therapeutic activity, but it basically was progressive disease. And the formulation was, seemed very simple and easy to describe. New graduate student steps into the picture. This is Devin. And he starts off on his thesis project, and he basically tries to reproduce what that group did. And he looked at BSA in combination with copper, and he looked at this other idea of adding copper to DDC on the outside of the liposome, just to see what would happen. My prediction, of course, was that it would just precipitate out, and it would be a useless product. But lo and behold, we ended up with copper-containing liposomes with DDC. The complex was inside the liposome, not on the outside of the liposome. He's since gone on to ask these questions about, is it the copper DDC that's inducing immunogenic cell death? And he's now going on to studies that are looking at whether this new formulation is one that is going to be useful in augmenting the activity of the agents. So here's the description of the different formulations. BSA, You can add it to copper gluconate. In the absence of BSA, it forms this precipitate. In the presence of BSA, it stays lovely in solution. You can put DDC and add it to copper-containing liposomes, and it loads. This is these graphs showing the copper DDC to lipid ratio as a function of time after addition. Everything looks great. And then you can add the copper DDC on the outside of the liposome. It goes in, and it also is associated with the liposomes. When you do it that methodology, it is no question that the copper DDC is with the liposome. And if you look at the lipid, which is the square levels, it co-fractionates with the circles, which is the copper DDC. All these formulations are very, very active in vitro. DDC has very little activity, less than 10 micromolar in vitro. But if you now add copper, you end up with a nanomolar active drug. So it's like 50 to 80 nanomolar. Well, this timing thing is killing me. This is just a formulation process. So we're not making a formulation that allows the copper DDC to stay in the circulation for a long period of time. It just changes something from that's not injectable to something that is injectable. And that's illustrated here in this lower figure, if I can do it. Liposomes stay in the circulation, but the DDC, which is these square lines, do not stay associated with the formulation. Same thing can be said about the BSA. The DDC that's associated with the BSA is rapidly eliminated from the circulation. So it's just a formulation. It is not a drug carrier. We're working on that. That's a different question. So this has gone on to efficacy studies, and one of the ones that we started looking at was in the context of immunotherapy, asking if you look at the therapeutic activity in an immune-competent animal versus an immune-compromised animal, one where ICD induction could lead to an improved therapeutic effect in the immune-competent animal, but would have no impact in the immune-incompetent animal, we could see something quite interesting. And just look at the square, the filled symbols here. So this is the copper DDC, where if you did studies in immune-competent animals, you saw a percent increase in survival of 70%. But if you did it in the immune-incompetent animal, you saw no improvement in survival. The difference between these two mice is really the fact that one has an immune system and one doesn't. So this is, for me, a very classic way of defining whether your drug has the potential to augment immune responses. So we've gone on to look at different markers, and I'm going to go through this. This is an animated study. So we look at the progression of cells. We look at the expression of the CRT, which is calreticulin on the surface, and that's using this in-cell flow cytometry-based assay. And we're really asking the question of what's the difference between DDC, copper, and copper DDC complex. And you can see in this case where you're using the copper DDC complex that you're seeing an improvement in expression of calreticulin on the surface. If we start looking at this idea of ATP release, this is done through a simple assay that we have in the lab, a microtiter assay that we have. And we can see again that it's not copper alone, it's not DDC alone, it's the combination of copper DDC which is causing this increase in ATP release. Our last marker, HMGB1, same thing, this is an ELISA-based assay. And in this case, it's the presence of both copper plus DDC, which is causing an enhancement of the HMGB1. So this was a pretty clear indication that this is inducing immunogenic cell death. Submitted the grant, it was rejected. This is an early phase of the grant. And one of the things that people came back with and said, well, copper is a ROS inducing agent, a reactive oxygen species producing agent, and other agents can do this. And they said, you really should be looking at other agents. And one of the ones they picked of was iron. And iron forms a complex lovely with the DDC, and that's shown by these color indications down here. DDC by itself is a clear solution. When it complexes a metal, it becomes colored and precipitates. So you start asking the question, and we just looked at one marker, ATP. It's only copper that's doing this. It's a very copper-specific event. And we looked at other ROS-inducing agents, which are not metals. And again, it did not induce this, the expression of these damp markers. So again, all of a sudden, it's the copper DDC that becomes the interesting item. And it's not copper iron or copper zinc or DDC, I shouldn't say copper, zinc, copper, DDC combined with zinc, DDC combined with nickel. None of those things worked. It was just the copper formulation. He's going on and asking further questions about, is it the copper DDC or is it copper? And one of the approaches that Devin used was just incubating cells with increasing amounts of copper to find out how it affected these damp markers, the ATP, HMGB1, tau reticulin. And in almost every case, we see a very nice correlation between more copper in the cell, more expression of these damp markers. So it really sort of implies that it's the copper that might be the interesting item here in terms of expression of these damp markers. Now, I hope everybody in the audience, I don't know how many people are in the audience, but those in the audience, I hope they're really thinking about how did that copper DDC liposomal formulation work when we added copper DDC to the outside of the liposome and it magically became inside the liposome. And it really is because DDC was acting as a copper ionophore. It was bringing copper into the liposome. And Devin asked the question of whether copper DDC was able to bring copper into cells. And that's what's shown here. I think this is CT26 cell, a colorectal cancer cell line. It was just looking at the level of copper in the cell as a function of time after adding the copper DDC. And you can see that it is very effectively increasing the level of copper inside the cell. So it's a copper delivery mechanism. And this becomes really interesting in the whole story. We have a bunch of different ligands that we know bind copper. Many of these ligands are interesting from an immunotherapy perspective and ICD induction. I point out mitoxantrone, a standard positive control, daunorubicin, a standard positive control. Some of these other agents, oxaliplatin, another positive control. The agents that really appeared most interesting to us were these agents that were known as ionophores. They were known by other people to be able to bring in metals into cells, like copper. And this one, copper cleoquinol, is proving to be particularly interesting, and that's going to be a future story. So let's move on to the sort of the end of the talk. I don't know how long I'm going on for, but this vascular normalization side of things, I'll do this very quickly. It's really asking whether this is going to be beneficial in the context of immunotherapy. And it's not a story that we started. It was a story that started ages ago from a therapy perspective. And I won't talk about that, but in the context of immunotherapy, Rakesh Jain demonstrated, this is a guy at MIT, he demonstrated that if you achieve tumor normalization, that you now can convert an immunosuppressive environment in a tumor to an immunosupportive environment. The idea that those normal blood vessels are better able to bring in immune cells that can mediate an immune response. And it's outlined here in terms of converting the immunosuppressive environment into an immunosupportive environment. The story around the ways to do this involved the use of Avastin, which is a VEGF. So this is vascular endothelial cell growth factor therapy, which was approved and used. So that's being combined with checkpoint inhibitors and this concept of metronomic dosing. So rather than giving a high bolus dose, you're doing a split dose. It's small, lower doses over time and asking for whether there's a therapeutic benefit. When therapeutic metronomic dosing was being used as a single agent, it was interesting because they saw therapeutic benefits in patients, but they reached something called a crossover point. All of a sudden during the trial, as the patients went on in months, there was a time when those patients that were getting metronomic dosing were actually doing worse than those patients that were doing conventional dosing. And it was believed that this was simply because of vascular normalization, that the vascular normalization created a more improved environment for the tumor cells to grow. So I think metronomic dosing as a single agent is not being worthwhile considering, but in the context of immunotherapy, it definitely is worthwhile considering. We got into this idea of vascular normalization through a formulation of a camptothecin called CPT1 or irinotecan. It's an interesting drug that is bioconverted to a different drug called SN38, which is much more active than irinotecan. It's 1,000-fold more effective. It's a drug that needs to be kept in a low pH environment, which is the way we try to encapsulate it and maintain it in a low pH environment, because if you're at a neutral environment, it forms this ring open structure, which is therapeutically inactive. We developed a formulation which involved the use of copper, of all things. You put copper inside the liposome, add irinotecan to the outside. Because irinotecan formed the complex with copper, you could all of a sudden get a formulation with irinotecan captured inside the liposome as a complex. And that formulation, we called it Irino4C, exhibited a much longer circulation lifetime and exhibited a vast improvement in therapeutic activity. The student that's been developing this particular program, Kevin Sun, has developed an improved technology around this, but we believe that those improvements, which are related to increased circulation lifetime, increased therapeutic effects, will also be best suited for use in the combination with checkpoint inhibitors, and that's what's being tested. The antivascular effect of Irino4C was established. We're convinced with this new formulation, we're going to be able to see the same thing, but the story goes this antivascular effect resulted in an increase in perfusion, which could be measured by a dye. This is Porsche 33342, and you could see in an Irino4C treated animal, you had more of this blue dye getting into the tumor. We also saw that where there's a change in the amount of endothelial, the CD31 positive cells. So we saw an increase in CD31 positive cells in the treated tumors, which again is a sign of tumor vascular normalization. We also saw a decrease in hypoxia. Hypoxia is there because the vasculature is poorly formed and it doesn't allow oxygen to distribute in the tumor well. But we actually could demonstrate in the irinotecan formulation treated animals that we saw a decrease in hypoxia, and we saw an increase in some of these anti-angiogenic factors. All signs of suggesting we're achieving tumor normalization. Finally, we looked at just movement of a small molecule. If you treat with a liposomal drug like Irino4C, and then you wait and treat with a small molecule, if you have achieved tumor normalization, you should be able to demonstrate that you're getting better distribution of that small molecule in the tumor. And that's exactly what we saw. And that's in this lower graph where we look at the amount of 5-FU delivered as a function of how many times we treated with the IRC formulation. Did that result in improved therapeutic effects? I would say this is a very complicated experiment. We would have to have a separate discussion on it, but it's quite clear that the combination of Irino4C followed by 5-FU at a time when we got tumor normalization, we actually saw improvements with 5-FU. And this is the basis of some of the studies that are ongoing now. We're really asking the questions of whether this is a drug that can be used in the context of immunotherapy. Can it be combined with a checkpoint inhibitor? Does it create an environment which improves the activity of the checkpoint inhibitor? And do you use this in sequential delivery, or do you administer stuff synergistically? But the goal really here is to take an improved irinotecan formulation and combine it with checkpoint inhibitors and move that forward into the clinic. So I'm going to end here. I think I've given time for questions, but Outline said immunotherapy for cancer is a game changer. Yes, it is. Immunogenic cell death, a way to kill cancer cells. It is. It's different. How can you take advantage of it is one of the things we're asking. Cuproptosis is a way of killing cancer cells, so that is a yes. And we are now thinking of copper as something that actually could be used as a therapeutic agent, perhaps as itself by itself, because it's forming a complex with a ionophore-like compound. This is another one of these ones where I'm constantly told we cannot develop this, but I talked to people that developed the platinum-based therapeutics, and they were told almost the same thing. You can't develop a toxic metal for treatment of cancer, but yet the platinums are now one of the standards of care for many different cancers. Tumor vascular normalization, it's a good thing, but probably not good when it's used as a single agent. But when it's used in combination with immunotherapy, it could be great. And everything we do right now is in the context of combinations. And we really are focusing on combinations in the context of immunotherapeutics. I think that's it. Hopefully I didn't do that too fast. The group is sort of outlined here. This is people, I think they're up at Grouse. Notice that I am not in this picture because I don't ski, which is probably a good thing. The group is small, but they're energetic and having a lot of fun. I've talked about data collected by Devin and by Kevin, and then the postdocs in the lab that are involved in some of these studies are Zinab and Miriam. We have summer students involved, and it's all supported by PharmaCore, which is Edmund funded. And that really is part of the investigational drug program, which is a team of animal care technicians plus people that know how to manufacture drugs so that we can get them into people. It's funded by NMIN, it's funded by CIHR. I've had previous funding from the Canadian Cancer Society, and again, BC Cancer Research Institute has been instrumental to allow this activity to occur. So I'll end there and address any questions that people have.
I think I noticed that there's one question already submitted, so I invite Louis to raise his hand and ask his question.
Thank you for the great presentation. And my question is, the first question I made about the middle of the presentation, so maybe you answered it already. It was regarding the susceptibility. If there is differential susceptibility to Cuproptosis from cancer cells and healthy cells.
Well, I'm going to say I don't know. I mean, we have looked at healthy cells. And to me, it's really trying to differentiate between dividing cell populations, which are typically cancer cells, and non-dividing cells. The Cuproptosis has been identified by other groups in the US. And I believe that method of cell death has really been focused on oncology indications. So I don't think people are looking at it in the context of normal cell populations. In the background of all this, and that goes, I go on about this because Celator had a very, very difficult time starting. It was partly because of copper toxicity. And the year that we started Celator was a year that a young woman in Alberta was killed by her friends. And it was because her friends used copper sulfate and mixed it in with a bubblegum flavored icy at 7-Eleven, and she drank it and died. So there was no question by anybody's mind that copper sulfate is toxic, and there's lots of evidence in the literature saying it's toxic, but it is at very high doses and when given orally over extended periods of time. So in the context of oncology, I think copper is a perfectly safe compound. And your second question, I'll just go to it. I touched upon this, Bob Kerbel and Judith Volkman led the idea of metronomic dosing to achieve vascular normalization, and they started instantly lots of clinical studies. And those clinical studies looked very promising at the start, but as the data rolled out, it looked like it was bad because it actually increased the viability of the tumor. And I think it is related to this metastatic potential. Because it created a more normal environment, it was a healthier environment for those cancer cells. And so the patients, for some of them, they actually did worse when they were on that metronomic dosing.
Thank you very much for sharing.
Mm-hmm.
I have a question, Marcel. The copper that's taken in, the copper that's active, do you have any idea if it's copper 1 or copper 2? I mean, I know it's copper 2 that goes in as in the salt in the complex, but of course it could be reduced to make copper 1. They have differential activities, right, towards ROS and stuff.
Yeah, absolutely. And again, I'm not going to be able to give you a clear answer. I think with the formulations that we're working with, it's copper 2. What's actually happening when we're delivering the material to the cells? I have to believe that it's complex. And again, is it complex as copper 1 or copper 2? I can't tell you.
I see. So maybe a technique to check for it would be an interesting idea.
Well, Devin's exploring some of these different ICP mass spec technologies, but I don't know if it's going to be able to allow us to differentiate between copper 1 and copper 2, especially inside tissue samples.
Yeah, there are these fluorescence lifetime experiments that can be done sometimes to differentiate the two oxidation states. the CRT, so you think there's more CRT being released from the ER, I guess. Is there more CRT binding to the surface? In other words, there can also be various endogenous, there can be other sources of CRT around that could bind on the cell that would then give rise to increased, phagocytosis, cell death, and so forth. Do you know that it actually comes from within the cell?
Again, can't speak to it. Our assumption is that it is from inside the cell. So we do talk about it in the context of mobilization of CRT from inside of the cell to the cell surface.
Right. Thanks.
Whether the CRT is coming from different sources, I can't speak to that.
Okay, thank you. I see a question from Ellen Wasan. Ellen, would you care to ask your question?
Hi, can you hear me okay?
I can. Hi, Ellen.
Hi. I'm just wondering what you think about the combination of using the Cuproptosis agents and bevacizumab with dendritic cell vaccines.
And CAR Ts, I would add to that.
Right.
I would say that it was not something that we had contemplated, but Kevin's been giving presentations in various venues to the immunotherapy groups here in BC Cancer. And there's a new person here, Laura, who has come from John Bell's group and is an expert in CAR Ts and immunotherapies. And she actually brought up this idea that, if you create this vascular normalization, that it might actually improve the activity of CAR T cells. It might actually improve the activity of some of these dendritic cell vaccines. So I have a feeling that is going to be one of the things that we're going to try, because we have an expert in the building that knows the CAR T area very well. So it's a very important question. And yeah, I like triple threat.
Are there trials going on with immunotherapies already with the bevacizumab?
Yes.
Okay.
I can't speak to which those trials are.
Mm-hmm.
Kevin might be able to do that better. It's an area that's a bit confusing. If you're dealing with an approved drug, it automatically allows you to go into clinical trials. So like I've mentioned this metronomic dosing, it was basically done with approved drugs. You would take a particular drug, pick a pink alkaloid, and instead of giving a big bolus injection every 3 weeks, you would give smaller injections daily for a week. The total dose was the same, but because the dose was lower over extended periods of time, they saw improvements in therapeutic activity. That was a research thing, but it went from research to clinical trial in a blink of an eye because they were dealing with an approved drug. And I think bevacizumab, as an example, is an approved drug, so they can actually go off and do the study. Whether it's the right study to do, I'm not sure. It is something that we have to address from a research perspective and maybe from a development perspective. If there are other agents that are already out there that induce immunogenic cell death, what are we bringing to the table? What's different about what we bring to the table? And I don't know if anybody's going to ask the question, but one of the things that you'll see with the Vyxeos product, which is a copper-containing product, where copper is complex with daunorubicin, and daunorubicin is known in the literature as an ICD inducer. That Vyxeos formulation was resulting in 30% long-term survival in the context of induction therapy for AML. People were commenting on the fact that this looked like immunotherapy. Again, you just ask, have to ask the question of whether it was actually inducing immunogenic cell death and whether that was actually causing this improved therapeutic output.
Yeah, many ideas. Unfortunately, we've come to the end of our time for today's lecture, but I'm not surprised if Marcel would answer questions by email and so forth subsequently. So I want to thank Marcel for his presentation, really showing us the power of drug combinations as opposed to single therapeutic approaches. So really, I'm a big believer in 10% greater probability of living a long time. Give it to me every time. And I invite and the attendees to please join me in thanking Dr. Marcel Bally for delivering this particular and wonderful March 2023 NMIN lecture.